Reproductive functions of the male can be divided into three major subdivisions. First, spermatogenesis. Second, performance of male sexual act. And third, regulation of male reproductive functions by the various hormones. Female reproductive functions can be divided into two major phases. First phase is preparation of the female body for conception and pregnancy, and second, the period of pregnancy itself. Today, we will focus on regulation of the male reproductive functions by hormones and preparation of the female body for conception and pregnancy. We will cover three topics. First, we will look at male sex hormones. This slide shows you four androgens. First three androgens have a double bond. Androstenedione and testosterone has a double bond at position 4. Dihydroepiandrosterone has a double bond at position 5. But in dihydrotestosterone, or DHT, the double bond is reduced. This reduction makes DHT the most potent natural androgen. So this is the first point. Second point, conversion from testosterone to DHT is catalyzed by 5-alpha reductase. This enzyme is found in target tissues outside the testes. Therefore, activation of androgen, especially testosterone, takes place in the peripheral tissues. The third point, testosterone is carried in the blood by albumin or sex hormone binding globulin. Testosterone binds more tightly with sex hormone binding globulin than with albumin. Androgen receptor is a nuclear receptor. It has a ligand binding domain, for example, as shown here, and DNA binding domain. After an androgen binds the receptor, two receptor molecules form a homodimer. And this is a DNA binding domain from one molecule, and this is a DNA binding domain from second molecule. These two form a dimer that binds DNA. Here is integration with receptor lecture. An androgen receptor is a type 1 nuclear receptor. Androgens are important for formation of male external genitalia, growth of prostate, male pattern baldness, and synthesis of nitric oxide synthase in penile tissue. Androgen also stimulate protein synthesis and decrease protein breakdown, especially in muscle. That's why, in general, men have more muscle than women. This slide is very similar to the one shown in the adrenal gland lecture. Very similar to synthesis of aldosterone and cortisol. The rate-limiting step is the first step, catalyzed by cholesterol desmolase. To differ from aldosterone and cortisol, removal of two carbons by 1720 lyase directs this pathway to androgen. Here is the testis, and this is seminiferous tubule. And here is a cross-section. When we zoom in, we will see Leydig cells lining outside of the tubule. Sertoli cells are epithelial cells that support germ cells development. Leydig cells bind luteinizing hormone and produce androgen. Sertoli cells bind follicle-stimulating hormone and produce androgen-binding proteins and inhibin B. Here we take a closer look at Leydig cells. And this is a Leydig cell. It receives signaling from luteinizing hormone. Activated luteinizing hormone receptor activates cholesterol desmolase. Androgen synthesis is increased. DHT and follicle-stimulating hormone then acts on Sertoli cells. Sertoli cells receive DHT and follicle-stimulating hormone and produce hormones and proteins involved in control of testes directly. Here are the hormones and proteins they produced. First one is anti-Mullerian hormone or Mullerian inhibiting factor or MIF. Androgen binding proteins. This protein increases the half-life of androgen. Sertoli cells also produce inhibin B for negative feedback. They produce estradiol to support spermatogenesis. In fact, the liver produces most of the male estrogen. Two stages in human development especially require androgens. One is fetal stage. The other one is at puberty. Fetal stage requires testosterone, DHT, and MIF. Testosterone is required for differentiation of Wolfram duct into efferent ducts. DHT is required for differentiation urogenital sinus into prostate. MIF is required for inhibition of female internal structure. Hypothalamus produces gonadotropin releasing hormone. It acts on anterior pituitary glands. The pituitary gland then releases two gonadotropins. One is luteinizing hormone that acts on Leydig cells. The other one is follicle-stimulating hormone that acts on Sertoli cells. Leydig cells produce androgens, including testosterone. Testosterone is converted to DHT in peripheral tissues. Testosterone and DHT have a negative feedback on hypothalamus and pituitary glands. The end result is decreased release of luteinizing hormone and follicle-stimulating hormone. Sertoli cells release inhibin B. Inhibin B has a negative feedback on pituitary glands to inhibit release of follicle-stimulating hormone. To sum it up, testosterone and DHT inhibit secretion of gonadotropin releasing hormone from hypothalamus. In addition, they also inhibit release of luteinizing hormone and follicle-stimulating hormone directly from anterior pituitary gland. Inhibin B inhibits release of follicle-stimulating hormone from pituitary gland. Now, let's move to part 2. In females, two types of ovarian sex hormones are estrogens and progestogens. By far the most important of the estrogens is estradiol. The most important progestogen is progesterone. Progesterone is derived from cholesterol. Estradiol is synthesized from testosterone. Like androgen receptors, estrogen receptors and progesterone receptors are type 1 nuclear receptors. As I described to you earlier, estradiol is synthesized from testosterone. The conversion is catalyzed by aromatase. There are three changes after the reaction. First, the ring A becomes aromatic. Second, reduction at the C3 position. Third, removal of methyl group at C19 position. In a normal non-pregnant woman, progesterone is secreted in a significant amount by corpus luteum only during the later half of each ovarian cycle. Female sex steroids are synthesized in theca cells and granulosa cells in ovarian follicles. Here is an ovarian follicle. These are theca cells, and these are granulosa cells, and this is an ovum. Theca cells express luteinizing hormone receptors, but not follicle-stimulating hormone receptors. They produce progesterone and supply androgens to granulosa cells. Granulosa cells initially express follicle-stimulating hormone receptors, but not luteinizing hormone receptors. They produce estradiol. Later in the ovarian cycle, granulosa cells also express luteinizing hormone receptors, and they start to secrete progesterone instead of estradiol. Luteinizing hormone receptors activate cholesterol desmolase to increase synthesis of progesterone. Follicle-stimulating hormone receptors activate aromatase to increase synthesis of estradiol. Hypothalamus secretes gonadotropin releasing hormone. It is a peptide hormone. Anterior pituitary glands express gonadotropin releasing hormone receptor. Activation of this receptor activates G-alpha-Q phospholipase C pathway. It is one of the 4 G-protein coupled receptors that utilize this pathway, as described before. The ovarian change that occur during menstrual cycle depends completely on these 2 gonadotropic hormones, luteinizing hormone and follicle-stimulating hormone. These hormones are absent during childhood, but the secretion of these 2 hormones increase at puberty. Luteinizing hormone and follicle-stimulating hormone are heterodimeric proteins made up of one alpha and one beta subunit. The alpha subunits of luteinizing hormone, follicle-stimulating hormone, TSH, and HCG are identical. Both luteinizing hormone receptor and follicle-stimulating hormone receptor are G-protein coupled receptors. Activation of this receptor leads to activation of cyclic AMP, protein kinase A pathway. HCG is produced by the embryo. It binds the same luteinizing hormone receptor. HCG has a cross-reactivity with thyroid-stimulating hormone receptor. A high level of HCG may cause some symptoms of hyperthyroidism in pregnant women. Sex hormones in females control physiologic changes that occur both in the ovaries and in the uterus during the menstrual cycle. The menstrual cycle can be described by the ovarian or uterine cycle. The ovarian cycle describes change that occurs in the follicles of the ovary. The uterine cycle describes changes in the endometrial lining of the uterus. Both cycles can be divided into three phases. Here is an ovarian cycle. These are gonadotropic hormone levels, and these are ovarian hormone levels. Ovarian cycle is divided into three phases: follicular phase, ovulation, and luteal phase. These represent a uterine cycle. It is comprised of three phases: menses, proliferative phase, and secretory phase. These are gonadotropic hormone levels, and these are ovarian hormone levels. And here is the follicular phase. In follicular phase, granulosa cells release estrogen and inhibin B. Estrogen has two effects. One is to act locally on granulosa cells to increase their sensitivity to follicle-stimulating hormone. Second is to act on anterior pituitary gland and inhibit release of luteinizing hormone and follicle-stimulating hormone. Inhibin B inhibits release of follicle-stimulating hormone from anterior pituitary gland. Again, these are hormone levels. Right before ovulation, estrogen levels rise. When they rise above a certain level, they no longer inhibit release of luteinizing hormone and follicle-stimulating hormone, as shown here. A rising estrogen levels instead stimulate the release of luteinizing hormone and follicle-stimulating hormone. Luteinizing hormone stimulate production of androgen, and follicle-stimulating hormone increase synthesis of estrogen. Eventually, this positive loop leads to surge of these two hormones. Especially luteinizing hormone surge triggers ovulation. Positive feedback loop is rare in the body. It only occurs at ovulation with estrogen and delivery with oxytocin. Now we are in the luteal phase. Starting in the later stage of follicular phase, estrogen along with follicle-stimulating hormone cause the granulosa cells to produce luteinizing hormone receptor. The metabolic pathways are altered to favor production of progesterone and reduced production of estrogen in granulosa cells. Finally, luteinizing hormone surge that occurs at ovulation drives both theca cells and granulosa cells to produce a large amount of progesterone. These cells are now transformed into luteal cells. Now, let's move to part 3. We are going to look at the clinical correlation. Let's first examine conditions that mainly affect males. The first condition is 17-alpha hydroxylase deficiency. Refer to the pathways for androgen synthesis that I have described to you earlier. 17-alpha hydroxylase deficiency blocks the synthesis of both cortisol and androgens. Especially insufficient androgen cause a disorder of sex development. It is a medical condition involving the reproductive system. Lack of androgen causes undervirilization in genetic males. They may show female external genitalia. Please note, the severity of this condition depends on the age of onset of deficiency. The earlier onset, the stronger the phenotype. These patients may have a history of hypertension due to elevated aldosterone levels. Second condition is 5-alpha reductase deficiency. It only affects genetic males. Although the phenotype can be variable, the affected males usually tend towards a female appearance. People with this condition are fertile with the ability to father children. The third condition is androgen insensitivity syndrome. It is caused by mutation in the androgen receptor gene. These pictures show you people with this condition. They are genetic males, but they are phenotypic females. They are unable to biologically reproduce. The fourth condition is Swyer syndrome. It is caused by a deficiency in one of the several genes on the Y chromosome, such as SRY gene. Very similar to androgen insensitivity syndrome. People with this condition are genetic males, but they are phenotypic females. In these people, gonads cannot make estrogen and progesterone. Breast will not develop, and uterus will not grow and menstruate until estrogen therapy is provided. The fifth condition is hypogonadism. Patients with this condition have diminished androgen biosynthesis. Very similarly, the severity of this condition also depends on the age of onset. Primary hypogonadism is caused by gonadal failure. Secondary hypogonadism is caused by defects in hypothalamus or pituitary gland. For example, hypothalamic pituitary disorders such as panhypopituitarism or Kallmann syndrome can cause hypogonadism. Kallmann syndrome is caused by gonadotropin releasing hormone deficiency. Hypogonadism may also be caused by Klinefelter syndrome or Noonan syndrome. Here is integration with genetics. Klinefelter syndrome is caused by presence of extra X chromosome. Now, let's look at conditions that affect females. The first condition is congenital adrenal hyperplasia. It represents a group of autosomal recessive disorders, each caused by a deficiency of an enzyme involved in synthesis of cortisol, aldosterone, or both. We will focus on two deficiencies. The first one is 21-alpha hydroxylase deficiency. It is the most common type of congenital adrenal hyperplasia. In these patients, mineralocorticoids and glucocorticoids are virtually absent, which causes hypotension. Overproduction of androgens causes ambiguous genitalia, excessive facial hair, virilization, and menstrual irregularities in these females. 11-beta hydroxylase deficiency has clinical presentations similar to 21-alpha hydroxylase deficiency, except that this condition leads to buildup of 11-deoxycorticosterone, which causes low-renin hypertension. Other deficiencies in the congenital adrenal hyperplasia are summarized in this slide. The second condition that affects females is Turner syndrome. It is also known as 45X. Patients with Turner syndrome have webbed neck and short stature at birth. Typically, they do not have menstrual periods and breasts without hormone treatment, and they are unable to have children without reproductive technology. Around 90% of girls with Turner syndrome don't produce enough of ovarian sex hormones. Here is integration with pharmacology. The birth control pill is also known as combined oral contraceptive pill. Includes a combination of estradiol and a progestin. Progestin is a synthetic steroid hormone that has effects similar to progesterone. Progesterone is a progestogen. Progestogen has negative feedback on hypothalamus to decrease release of gonadotropic releasing hormone, which leads to a reduced level of follicle-stimulating hormone. As a result, there will be no increased level of estradiol. An estradiol luteinizing hormone positive feedback loop is disrupted. At the end, there will be no luteinizing hormone surge, and therefore no ovulation. Estradiol has negative feedback on the anterior pituitary gland to decrease secretion of follicle-stimulating hormone, which also disrupt luteinizing hormone estrogen positive feedback loop and prevents ovulation. Here are hormonal changes in some conditions. For example, primary hypogonadism is caused by gonadal failure. As a result, release of sex steroid is reduced. Negative feedback on pituitary glands does not occur, and level of luteinizing hormone, follicle-stimulating hormone rise. Use the same idea. Students continue to fill in the rest of this table as your homework. Another exercise. Given changes in sex steroids, gonadotropic hormones, we need to tell what conditions these hormonal changes are related to. For example, when we encounter a condition in which all three levels are down, this situation most likely occurs when there's a failure in hypothalamus or pituitary gland. Two strong candidates are pituitary hypogonadism and Kallmann syndrome. Use the same methods. Students continue to fill in the rest of this table as your homework.